Okay, Assalamualaikum and good afternoon, everyone. I hope you are doing good. Sorry that we have to do replacement class at this time. Okay, but I think we really should do so that you can go for your holiday peacefully next week. Okay, so today we will look into how exam questions will be structured. Okay, we will revise a bit on all of the pathways that we have gone through in biochem. Okay, as a recap. Okay, so okay. For your final exam structure, again, I will go through this. We have two parts. Part A is the MCQ questions. Okay, total question is 25 and then 1 mark per question. And then it will cover chapter 1 until 14, basically all. And you need to answer all questions. So that is for MCQ questions. The level is basically knowledge, okay, meaning that you really need to read for you to answer the questions. Okay, and part B is essay questions. Okay, we are given 5 questions, so each will carry 25 marks. You need to answer 3 out of 5 questions. Okay, you can read through the instructions in your final exam question later. Okay, and for your essay questions, it will cover chapter 7, lipid metabolism, okay, to food biochemistry. Okay, so the 5 questions will be either of the chapters from 7 to 14 for the essay questions. Okay, so I have go through this with you in the previous tutorials. Okay, this is the structure of your essay for your answers. Okay, always remember to write in an introduction. Don't go straight away write your answers for the questions. Okay, start with an appropriate introduction. You can rephrase the question back, or you can even write in, for example, if the question asks you on lipid metabolism, so you can define lipid, for example, says the function of lipid, okay, on the introduction. Okay, and then make sure you have a conclusion. You can summarize all the points, what have you written, and there's also marks in terms of your flow and organization. Okay, and the remaining marks would be for your content. Okay, so that is your final exam structure. So let's briefly recap this. Okay, so it would be helpful if you have this table with you that I have asked you to do in previous class. Okay, the summary table. I have asked you to do the carbohydrates metabolism with these aspects. Okay, what you can do later when you do your revision, add on the other metabolic pathways. Okay, like for protein metabolism and lipid metabolism. The same thing, write in the definition, purpose, location so that you can better memorize them. Okay. So in order for us to summarize and to integrate all the pathways that we have learned in biochem, we need to understand these two conditions, the well-fed state and the fasting state. Well-fed simply means you are being fed after you have eaten or taken food. So in well-fed state, the key point here is to look into glucose concentration in your blood. Okay, so we will start first with fat state. Okay, so in fat state, remember it's after you have taken a meal. Okay, so first thing, the key point is glucose concentration will increase in your blood. Okay, so when glucose concentration increase, who will detect this is this super sensitive organs called pancreas. Okay, make sure you are familiar. You go through back the definition of these terms, because in your presentation before this, some of you mix up all the terms. This is hormones lah, organs lah, all of these are mixed up. So make sure you go through back, okay, the definition. So again, glucose is a molecule. Okay, so there's an increased concentration of glucose. So who will detect? Pancreas. Okay, pancreas is an organ. Okay, so what will pancreas do after detecting a rise in glucose concentration? It will release a hormone called insulin. Okay, so insulin is a hormone. Okay, so what will insulin do? It will activate these 4 pathways here. Okay, so in fat state, what will be activated or increased is glycolysis, glucogenesis. Sorry, this is glucotypo. Glucogenesis, lipogenesis, and protein synthesis. Okay, let's look into one by one. Okay, so if you remember, glycolysis, glyco is the glucose, okay, lysis is the breakdown. So in glycolysis is the breakdown of glucose into pyruvate. Okay, why glycolysis takes place in fat state? Go back to the key point earlier, because we have a lot of glucose. Okay, in fat state, the glucose concentration increase. So we need to bring down or break down the glucose that is high in our blood. So that's why in fat state, glycolysis is activated. Okay, okay. So next is what is activated is glucogenesis. Okay, so glucogen, nesis is the produce, okay, synthesis. Okay, so in glucogenesis is when we are trying to store glucose into, converting glucose into glycogen. Okay, so why does this happen? Again, go back to the point of fat state, there's high glucose. So now we have broken down the glucose in glycolysis. So the excess glucose, what should we do with them? Is to store them in the form of glycogen. Okay, so that's why glucogenesis takes place. Okay, to store excess glucose. We have a lot of glucose, so we have to store them. Okay. So next point, what happens in fat state is lipogenesis, or we don't cover this in our syllabus, but just for you to understand lipogenesis. Lipo is the fat tissues. Genesis means to form. Okay, so remember, whenever we have eaten something, we don't only eat something from the carbohydrate sources. Okay, we also take things from fat sources, for example. Okay, so in lipogenesis is basically the formation of fatty tissues or adipose tissues. Okay, that's why the more we eat, the easier for us to get fat. Okay, so that is why lipogenesis takes place in fat state. That's how you remember it. Okay, so the next thing is protein synthesis. Okay, basically to form, build up our skeletal muscle, okay, our enzymes, our transport proteins, for example, okay, from the dietary protein that we have eaten. Okay, so there's a protein synthesis taking place to build up or to repair our tissues. Okay, so let me quickly go through. Okay, fat state meaning after we have eaten something. After we have eaten something, of course, our glucose concentration will increase. So who will detect this? Pancreas. Pancreas is an organ. So pancreas will release insulin. So insulin is a hormone. Hormone means it's a signaling molecule. It will send signal to our body. Okay, so insulin will send signal. Okay, now we have high glucose concentration. So what our body will do upon receiving the signal from insulin, it will kickstart glycolysis, glucogenesis, lipogenesis, and protein synthesis. Okay, to manage the high glucose concentration and also to manage whatever we have eaten based on fats and also protein, to utilize them for energy, for example. Okay, so that is fat state. Are you clear? Do you have any question on fat state?
No.
Okay, so that is fat state. So let's look into fasting state. fasting meaning you have not eaten for some time or after a vigorous exercise. Okay, so again, the key point, the indicator is the glucose concentration in our blood. So fasting state is the opposite of fat state. Okay, so in fat state, we have increase in glucose concentration. So in fasting state, it's about the decrease of glucose concentration in our blood. So again, who will detect this? The sensitive organs called pancreas. So if in fat state, pancreas will release a hormone called insulin. So in fasting state, pancreas will release a hormone called glucagon. Okay, so glucagon, don't be confused, this is a hormone. Okay, so again, glucagon is a type of hormone. Remember, hormone is a signaling molecule. It will send signals to our body to kickstart a metabolic reaction. Okay, so what kind of metabolic reactions will glucagon trigger or activate is this: gluconeogenesis, glycogenolysis, lipolysis, and proteolysis. Okay, so in fasting state, again going back, the glucose concentration is very low. So our body is trying to top up the glucose that is reduced. Okay, bring up the glucose level back to normal. Okay, that is why the first, this is not in sequence, this will happen concurrently. Okay, okay, so in fasting state, gluconeogenesis will take place. So what is gluconeogenesis? So gluco is the glucose part, neo is the new, genesis means to produce. So gluconeogenesis is to produce more glucose, but remember, gluconeogenesis is about producing glucose from non-carbohydrate sources. Okay, neogenesis, gluconeogenesis, meaning it's producing carbohydrates, okay, sorry, glucose from amino acid and also lipids. Okay, so gluconeogenesis takes place. Why? We are trying to produce more glucose. Why? Because in fasting, we have reduced glucose concentration. Okay, so that is gluconeogenesis. Next, we have glycogenolysis. Okay, so glycogen is the glycogen part, lysis is the breakdown. Okay, so remember, glycogen, let me draw. Glycogen is the storage form of glucose. Okay, glycogen is the storage form of glucose. So why glycogenolysis takes place in fasting state? Because we are trying to break down the glycogen to retrieve the storage of glucose back. Okay, so now we are trying to convert glycogen back to glucose. Okay, why? Because again, we have reduced glucose concentration in our blood. So glycogenolysis will take place in fasting state. We are trying to produce more glucose. Okay, as I explain this, I think you will get the importance of the table that I showed you just now. Okay, so that you will have an overview what are these reactions producing. Okay, what are the end products of these metabolic reactions. Okay, we also have lipolysis. Okay, if in fat state we are forming adipose tissue. Okay, so in fasting state we are trying to break down the adipose tissue or the fat tissues, which it will eventually produce ATP, energy. Okay, so remember, in fasting state, you don't have energy. You have not eaten for some time. So you will need energy. So where will this energy come from? One of it is from lipolysis, the breaking down of our adipose or fat tissue. Okay, and then this lipolysis also relates to this one, gluconeogenesis. Okay, what we call as the intermediates. Okay, lipolysis will have its own pathway, and within the pathway, it will keep producing some intermediates molecule. Okay, those molecules will be used for gluconeogenesis. Okay, to produce more glucose. Okay, we also have proteolysis. Okay, same goes to lipolysis just now. It's the breaking down of proteins. Okay, and proteolysis is also used for gluconeogenesis to produce more glucose. Okay, up until that, do you have any question?
No, doctor.
Okay, anyone else? Okay.
No, doctor.
So it's important for you to understand which metabolic reactions occur at which state. Okay, in fat state and also fasting state. Okay, so sometimes questions can ask you not that straightforward. Okay, for example, it will give a situation where it will ask, okay, insulin will activate which of the following reactions, for example. Okay, so that's why it's always important to go back to this diagram. Okay, for example, okay, let me give you a situation. If you have high insulin concentration, okay, so high insulin concentration, does it indicate high glucose or low glucose when we have high insulin?
Low glucose, doctor.
Low glucose ke? What causes insulin to increase? Go back to this diagram.
High glucose triggering the secretion of insulin.
Okay, okay. So always go back to this diagram. Okay, when there's increase in insulin, okay, who will release insulin? Pancreas. Okay, so what causes pancreas to increase insulin? Because pancreas is detecting our body have high glucose concentration. Okay, so if question asks you percent high insulin level indicating what? So it must indicate that we have high glucose concentration. Okay, so high insulin, it will activate these metabolic reactions, glycolysis, glycogenesis, lipogenesis, and protein synthesis. Okay, but insulin, the presence of insulin will inhibit the reactions within fasting state here. So that's why I put here plus symbol, okay, and this is negative, okay, or the inhibition symbol. Okay, so high insulin will inhibit glycogenolysis. Why do you think high insulin will inhibit glycogenolysis?
Because the excess glucose is converted into glycogen.
Yes. So high insulin again indicates high glucose concentration in our blood. Okay, but glycogenolysis is about producing more glucose. Kan over here. This one, sorry. Okay, producing more glucose. Okay, so we don't want to increase more glucose in our blood. We are trying to utilize the glucose. So that is why person who are having, when we have high insulin level, it will inhibit glycogenolysis because we don't need more glucose concentration in our blood, but we need to convert the glucose or break down the glucose for us to utilize the energy. Okay, same goes to high insulin will inhibit gluconeogenesis. Why? Because high insulin indicates we already have high glucose. Okay, so we don't need more glucose to be produced from non-carbohydrate sources. Okay, same goes to glucagon. If we have high amount of glucagon, again, who will secrete or produce this glucagon is pancreas. Okay, so why pancreas decide to produce glucagon? When pancreas detect our blood is having low glucose concentration. Okay, so that is why glucagon will inhibit glycolysis, because glycolysis can only happen when we have glucose. Okay, but in fasting state, where glucagon is released, glucose concentration is very low. So we cannot break down more glucose. Okay, we need to produce more glucose. Okay. So does this makes you understand all this pathway integration better, or did I just scare you?
Paham, doktor.
Faham Orang lain, can you give me some reactions and response? Does it make this clearer or you are more confused now before we go into the questions?
Yeah,
Okay,
it's
doctor.
okay.
Okay, I will give you some time to digest. Let's take 1 to 2 minutes break and we continue to discuss the essay questions. Okay, I will end the session and come back to the same link, okay?
Okay, doctor.